All done.
Great. So welcome out to this webinar series on microglia. I'm Guy Brown from the University of Cambridge, and my co-host is Claire Butler. And so this webinar series consists of monthly talks of about 45 minutes by European scientists talking about their research on microglia, followed by a question and answer session. And today's talk is by Adam Dennis from based at the Hungarian Academy of Sciences in Budapest, and he'll be talking about how microglia shape neurovascular responses via compartment-specific mechanisms. So during the talk, please keep your microphones and cameras off to preserve bandwidth. And during the talk, if you think of a question, please put it into the chat function and direct it to all. And at the end, we'll read out those questions, but please do the questions during the talk, so there's not a great rush at the end. Okay, I'm going to now hand over to my co-host, Claire Butler, who's going to introduce the speaker.
Great, thank you, Guy. So yeah, a little bit about Adam, some background. So Adam's main interest is in neuroimmunology, neuroinflammation, with a main focus on microglia. In 2005, he came to Manchester to work with Stuart Allen and Nancy Rothwell under a Marie Curie fellowship. And after finishing his PhD in Hungary, he returned back to Manchester to lead research on an EU-funded program. Then he returned back to the Institute of Experimental Medicine in Budapest, building his own lab from 2013, which was supported by the Hungarian Brain Research Program, among others. He was recently, in 2017, awarded an ERC Consolidators grant to understand how microglia shape neuronal activity and injury in health and disease. Adam's lab have recently published some really interesting papers on this topic, and I can send a few of these links through when I send the recording of the seminar at a later date. So with that, I'd like to give the floor to Adam. Yeah.
Thanks a lot for this very kind introduction, and it's really a pleasure for me to speak about our work. So basically, I would like to start how actually we came to study microglia. It happened approximately in 2001 when we had a crazy idea to map how the central autonomic nervous system controls the activity of immune organs, including the bone marrow. And to this end, we injected retrograde pseudorabies virus, which can move transsynaptically into the brain from peripheral targets, and basically visualize viral infected neurons, and it gives you all the connections which are synaptically linked and connect the higher order nuclei in the brain with the target organ where the virus was injected to. And surprisingly, apart from studying this very exciting pathway, we also noticed that viral infected neurons disappear over time, and basically these viral infected neurons from the early infected regions, such as the medulla, start disappearing as the viral infection propagates toward the cortical areas. And we were also lucky because I had a colleague who genetically engineered these viruses at the time, Zsolt Boldogkői. So we had an immediate early GFP marker to look at early infected neurons, and another marker for the viral structural proteins. So we could kind of match the infection times for individual neurons, and found that very weird looking lectin positive cells are recruited around these neurons, as we learned from in vitro studies already a few hours after they become infected, and these turn out to be microglial cells. Now, it took approximately 15 years to repeat this experiment with red pseudorabies virus strain, directing the infection into the cortex and find out that these microglial cells are really recruited in real time to these infected neurons. And what you can see is small particles of the neuron as the microglial cells dislocate with their cell body in this case, and they rip the neuron into pieces. And you can see this barrier-like structure being formed around the infected neuron over like 2 or 3 hours. Certainly, it looked very exciting, and we had also other interesting observations in the meantime. Among them, we did some research in Manchester on microglia, and found that microglial cells change quite substantially in response to certain systemic inflammatory conditions. We did it with Nancy Rothwell and Stuart Allen. This is just a clinical study combined with experimental data showing that in patients with multiple risk factors for stroke who have chronically elevated CRP, there is an increased TSPO binding indicative of increased neuroinflammation in the brain. And if you get the same model or similar model in a so-called corpulent rat, which are obese, atherosclerotic, and have multiple conditions as they get older, you can see that TSPO binding also increases in these animals without hurting them with any other means, indicating that microglial function changes over time, and it might have some implications to disease. Certainly at that time, it was not very easy to manipulate microglial cells. And I was very much interested in how these really broadly known inflammatory conditions seen in almost all brain diseases are influenced by microglial activity and responses. If you look at the model, how the field used to imagine, and still some people would imagine microglial function in these conditions, we have this kind of resting or surveilling microglial cells, which are or appear to be important in brain development and maintaining normal brain function. And then something happens. It could be an acute stimulus or chronic inflammatory changes. And then these microglial cells become activated. They start phagocytosing multiple things like dying neurons or synapses and produce inflammatory mediators. And then it would explain why neuronal injury develops. Now, the problem with this model, as it was relatively obvious from our own studies, but also from others in the field, that if you look pro-inflammatory mediators in many models, such as after experimental stroke, it's mostly protective, whereas microglial activation and interfering with microglial activation has all sorts of phenotype. And one of the conclusions, among many possible others, which I drawn, was that we actually don't have really good tools to manipulate microglia. And in fact, we didn't really have these tools until very recently. I just want to tell you, or want to show you a very simple example to this. Basically, we have this well-known paper from Richard Ransdorf groups on the left-hand side with the microglia gain of function mice, these fractalkine receptor knockout animals. And you can see that if they induce an MPTP based Parkinsonism model in them, then increased neurodegeneration is seen. And we got the mice in 2006, soon after the paper was published, and found that after experimental stroke, injury is smaller. So in other words, you find these models very useful to implicate microglia to different pathophysiological responses, but I found it very difficult to tell exactly what these microglia who have no fractalkine receptor are doing. And I was very lucky to make contacts with an American company, Plexicon. Probably many of you know them well by now, but in 2015, they were not that much recognized for microglia biology. And they developed a series of inhibitors CSF1 receptor kinase inhibitors for synovial tumors, and found that if you feed this to normal animals over a few days or weeks, basically peripheral macrophages are retained, but microglial cells disappear from the brain. And what is really important, they die as a result of apoptosis. So you don't really have BBB breakdown, you don't really have inflammation, kind of a silent way to remove microglia from the brain. And I found this tool really powerful to study how microglial cells actually contribute to different brain conditions without causing inflammation. This is just an experiment from our 2016 paper. If we feed this PLX drug, 3397 in this case, mice for 3 weeks, you can see that we can get rid of almost 95–97% of microglia. If we withdraw the drug for a day, microglia remain depleted, but if we do the drug for 2 weeks, microglia are repopulated. And if we perform experimental stroke, surprisingly, brain injury becomes larger. It's the same if the drug acutely is not present, but if microglia return to the brain, then injury becomes very similar to that of control animals, showing that having no microglia in the brain, at least after acute brain injury, is not very useful. How can this work? How would microglial cells interact with injured neurons? So we did calcium imaging and try to co-register how microglial process dynamics works alongside with neuronal intracellular calcium changes. You can see this in the calcium in kind of whitish color, and microglial processes in green. And they found that basically when after experimental stroke in the penumbral tissue, where there are apparently still viable but injured neurons, when intracellular calcium went up, microglial processes got closer to the neuronal cell body, and they were withdrawn when calcium went down. If you deplete microglia and do the same imaging, you can see that neuronal network activity changes substantially. You see very weird oscillations coming, and microglia over time pick up much more intracellular calcium if you accumulate it, and you could also see that these cells are dying quicker, explaining, at least to some extent, larger brain injury seen in the absence of microglia. Now, how can microglial cells interact with neurons? In the last decade or so, primarily researchers explored the interactions between microglia and synapses, and certainly there were other forms mentioned or visualized, but there was no mechanistic information apart from a few isolated fields. And there are a few issues to consider, considering interactions and how they might function in real time. One is that neurons have very stable compartments, and this is the somatic and perisomatic compartments, while we see a high turnover of synaptic boutons and dendritic spines, which would really make it difficult for microglia to monitor neurons over a long period of time. Second is that the majority of mitochondria, which are really critical for cell-fate decisions, and they are also like immunometabolic hubs, are localized mostly in the soma. And also microglial synapse interactions often occur at very great distance from the injured neuronal somata. And it was very difficult to imagine how microglial cells could really interfere with this. And if you look at very closely neurons and microglia, this is a Kv2.1 staining for neurons, you can see that quite a few perisomatic compartments are touched by microglial processes. And certainly we were really excited about whether there might be any functional specialization at these areas, or this is just a random scanning of anything in the brain by microglial processes, and soma would be one of them. And two very talented colleagues of mine, Csaba Cserép and Balázs Pósvai, reconstructed neurons in 3D, visualizing all the perisomatic synapses, GABAergic and glutamatergic terminals, and microglia, and also the neuronal cell body, and found that microglial processes really touch these neuronal cell body regions where there is no apparent perisomatic button in the processes, in the vicinity of the processes. And this was the same in the human brain, indicating that there might be something interesting to study here. And moreover, we found that 80% of neurons received some sort of microglial process contact around their soma. If you look at synaptic contacts, visualizing GABAergic and glutamatergic synapses with VGAT Gefrin and VGLUT1 Homer respectively, you can see that these contacts appear to be less frequent in perfused fixed brain tissues. And in the human brain, it's the same, including the frequent contacts between microglial processes and the neuronal cell body. I can show you the somatic contacts between neurons and microglia, and you can see that these microglial processes really do scan around specific area of the neuronal cell body, and they also return to the same site. So probably if you look at this arrow on the left-hand side, you can see that there is a contact being formed here, and then it disappears and then comes back again. And what we found, surprisingly, that the lifetime of these somatic contacts is much longer. It's approximately 30-35 minutes compared to the average lifetime of dendritic contacts, which would be something around 7-8 minutes. Further suggesting that these microglial processes may be doing something to the neurons. Now, how can we visualize this in fixed brain tissue to make it very specific for microglia? We used P2Y12 immune fluorescence, and P2Y12 receptor is quite specific for microglia in the brain. I mean, if you look at transcriptomic studies, I mean, there is some low-grade expression in other cell types, but if you do immune fluorescence, immunogold staining, or immunohistochemistry, you do see that actually in the normal brain and in many conditions under injury, microglia, P2Y12 is quite specific for microglia. And certainly the staining goes away in knockout animals, further indicating that it might be a good tool to study where these microglial processes actually touch neurons. And we did electron microscopic examinations, found that as opposed to other markers, this P2Y12 is not just sitting on the membrane, but seemingly it forms clusters in some sites when the microglial process appear to touch neuronal cell body. And then we wanted to quantify it, so we turned to super resolution microscopy, which can visualize these P2Y12 positive small clusters at 20 nanometer lateral resolution, and found that on the same microglial cell, if a process touches a neuronal cell body, clustering of P2Y12 receptor increases sometimes almost twofold at these sites as compared to the non-contact areas. And we found this very similar in excitatory neurons and also in different populations of GABAergic cells. So how can this work? This was a typical time when we turned to other methodologies, because we really wanted to understand the foundations. How could these microglial processes find exactly these sites at the soma, and whether these sites have anything specific compared to other parts. But you can see that it is an immunogold visualization of P2Y12. It gave also support that perisomatic buttons didn't appear to recreate a lot of these clusters, but there was quite a dense staining on the somatic interface. And then we did some electron tomography. And then these 3D reconstructions really showed something very exciting, kind of a new structure on the somatic side of the neuron. So you can see the magenta is the neuron, and there is always a closely anchored mitochondrion very close to the membrane, some really weird intracellular membrane structure with vesicle-like things, with all these membrane enrichments down to the membrane, kind of anchoring this mitochondrial like structure or interface to the membrane, and the opposing green microglial process gets very close, and exactly where it gets really close to the neuronal membrane, you can see the enrichment of P2Y12 clusters at these sites. And when you quantify this, you can see that really it is not a clustering around the buttons, but it is a clustering at the somatic interface, very specifically to these sites. Now, what can be on the neuronal interface which would recruit microglial processes there? And it was a complete chance to look at Kv2.1. We used it first as a neuronal marker, but then we found out from the literature that actually Kv2.1 does not really function as a channel in many of the neurons when it's clustered, but it does function as a kind of exocytotic hub, anchoring all the apparatus to the neuronal membrane, which is responsible for exocytosis. And multiple papers pointed to the same direction, implicating these sites as really hotspots on the neurons for exocytosis and endocytosis and cell adhesion. Now, then we looked with double storm and found that excitingly, in many cases, these perisomatic sites contacted by microglia show co-clustering of Kv2.1 and P2Y12. And when you deplete microglia, these Kv2.1 clusters remain very similar, suggesting that probably this is an evolutionary feature of the neuron rather than something induced by microglial contact. And microglia learn to recognize these specific sites where exocytotic processes are frequent. To understand this a little bit better, we went on and try to find quantitative information where these clusters would really colocalize with increased Kv2.1 fluorescent density, and this is the case. So an unbiased analysis shows you very well that really microglial processes go to these clusters. And then we go in vitro and ask whether these clusters would recruit microglial processes. So you can use hex cells which don't have Kv2.1 or Kv2.2 channels, and you can transfect them with a normal GFP Kv2.1 plasmid, or you can transfect them with the dominant negative form, which cannot go into the membrane, and then can scan whether these microglial processes would make a more frequent contact with either the hex cells membrane or the clusters. And indeed, this is the case. The dominant negative plasmid really kills this frequent contact between microglia and the hex cells. And I can show you the video. You can see on the left-hand side this microglial process really, and also on the right-hand side of the image or the video goes really down directly to these clusters. What else can be expressed here? We found also that these very closely anchored mitochondria do appear more frequently in the contact area than outside the contacts. And also we found VNOT, vesicular nucleotide transporter, enriched here, which indicated that it might happen that ATP gets packed into vesicles and it is released at the specific hotspots of the neurons. And this could be something microglia could recognize. How can we prove this functionally, whether neurons can release ATP? So you can culture mature neurons, depolarize them with multiple ways, in this case 40 millimolar potassium chloride, and then it is possible to look how actually it would induce ATP release. And then you find that ATP is not only released here, but when you apply a kind of synaptic cocktail, as we call it, to inhibit release of synaptic vesicles, not much happens, as opposed to the L-type calcium channel blocker, nimodipine, which would be more something associated with somatic exocytosis. and also the same thing happens after very, very low dose of chloridronate, so it does not hurt microglia, which is a VNOT inhibitor, implying that really at these somatic compartments, ATP gets released. And we went on to visualize this. So quinocrine fluorescent labeling really nicely visualizes these ATP-loaded vesicles. And if you depolarize neurons in multiple ways, you can see that there are specific hotspots. These look like vesicles or large or even multivesicular bodies where this quinocrine labeled ATP containing vesicle disappears after neuronal stimulation. And since then, we've done all sorts of other things, and now visualizing these interactions with ATP sensors as well. So ATP is not a primary ligand for P2Y12, but ADP is. And another question was, would we have ecto-ATPases directly expressed on microglial processes which would cleave ATP on site? And actually, this is the case. You can see it with immunofluorescence and also with electron microscopic detection that at the junctions you do see enrichment of ecto-ATPases. Now, so we kind of found a really exciting looking structure, a new structure with closely anchored mitochondria, vesicular nucleotide transporter Kv2.1 channels, and clustering of P2Y12. But certainly nothing at that time was known about the function. So we first wanted to explore whether if we block P2Y12, would this somatic contact or somatic junction frequency decrease, and this is exactly the case. As you can see here, basically we can nicely reduce the contact lifetime in vivo during 2-photon imaging if we block P2Y12 by injecting PSB 0739 into the cisterna magna, but we didn't really touch those dendritic contacts. Now, you can turn it around and try to activate neurons with threads in a chemogenetic model. This is very simple because we can inject a virus into the neocortex expressing the dead receptor and can trigger activation just by injecting these mice intraperitoneally. You can see that these neurons are activated as also shown by increased CFOS expression. And when we basically look how microglial processes respond to this neuronal activation, you can see that the microglial process coverage increases, and it does not happen in the absence of functional P2Y12, suggesting that actually P2Y12 mediated recruitment of microglial processes to these somatic junctions and increasing coverage is really an active process, and it requires functional P2Y12 signaling. Now, then with the help of Gábor Molnár and Gábor Tamás, we turned to an ex vivo model. This is the only experiment we couldn't really do in vivo, cutting brain slices, and we really wanted to see whether the contacting microglial processes would have any interaction with neuronal mitochondria by visualizing NOT-H fluorescence in the neurons. And you can see whenever we have a contacting microglial process, we really see really an increase almost at the same time in NOT-H fluorescence, and this increase does not occur if we use brain slices from P2Y12 knockout animals, implying that there might be bidirectional signaling between microglial processes and neurons at these sites, and this would allow microglia to sense and hopefully influence neuronal mitochondrial function. And then we turn to the stroke model again, and we established quite a few different methodologies to image penumbral neurons, which are really hit by ischemia, but they are actually still functional, struggling for life, and we could then see how these contacting microglial processes interfere with these actions. You can see that in the penumbral tissue, we do see mitochondrial fragmentation in these injured cells, and also we see declustering of Kv2.1 channels, which has been known for different physiological and pathophysiological neuronal responses, especially during really strong depolarization. And then when we ask the question, what happens if microglial processes cannot really interfere with this, we found that first, microglial process coverage increases around neurons in the penumbra where Kb2.1 declustered, and second, we found that if we block these interactions by blocking microglial P2Y12 with the same approach as seen earlier, we can prevent this increase in process coverage. And as another functional readout, we also turned back to calcium imaging. We also did some functional conductivity measurements. This latter with laboratory of R2 Lees in Munich, and this present experiment in Hungary in my lab, and found that basically if you block acutely microglial P2Y12, excitotoxicity increases in this penumbral tissue, and this is associated with an increased infarct size and also worse neurological outcome. Another thing I would like to point out here, why these somatic contacts could be interesting even beyond stroke. This is a paper when we looked how microglia control the spread of virus infection in the brain. And you can see that these contacting microglial processes get very close to the somatic membrane even prior to phagocytosis, and there are certain interesting anchor structures which might be integrins or other proteins. This is being explored. So we also suggest that these sites, these somatic junctions, are ideal starting points for phagocytosis, because this way microglia would have a possibility to kind of record neuronal activity changes and injury-related signals, superoxide or ATP and other danger molecules released from the neuron. And if the neuron is terminally injured, this is already a very good anchor point to start phagocytosis. So kind of this area is still being researched intensely, and we hope to serve some other explanation soon on that. So just to sum up, we have an interesting structure on the neuronal soma with really unique ultra-structural phenotype and marker expression. And this is a site where microglial processes can actively be recruited and increase their process coverage in response to neuronal activity. This is P2Y12 receptor dependent. This is through which microglial processes can interfere with neuronal mitochondrial responses, at least sense mitochondrial readout, in particular sense ATP release, and supposedly can feed back to neuronal activity, as shown by a few studies. And we believe that this form of communication could be very exciting in many brain diseases, considering that the contacting microglial process around the soma will make approximately 500,000 contacts in 10 years' time, which is quite a lot. So,
Martín.
Yeah.
Papá tiene que ir a la farmacia ahora. ¿Tú quieres ir con papá?
Okay, so I try to continue, but I could see someone speaking at the back. Okay.
Sorry, can anybody else on this webinar turn off their microphones now?
Yep, I've muted whoever that was. So
Okay.
yeah, carry on, Adam.
Thank you. So, and then I would like to just very briefly show you a few other things. First, we, in a recent review with Csaba Cserép and Balázs Pósroy, tried to bring together all the information available about the contactomics between microglia and neurons. Certainly, the hotspots are the microglia synapse, the somatic junctions, and the axon initial segments, among many other things. And as you can see, actually, the localization of really key proteins and receptors, which are well known to shape microglial responses, neuronal responses, and microglial neuronal interactions in broad sense, is not really thoroughly done, to be honest. So there is a lot to explore about the compartment-specific details of these proteins and how actually we should think about these very, very specific sites, for example, the axon-industrial segment, where you can really interfere with neuronal depolarization, for example, or how would microglial processing make these really sophisticated decisions to eliminate a synapse or not through complement-mediated mechanisms.